My name is Amanda Smythers, and I'm a second-year graduate student in the laboratory of Leslie Hicks in the Department of Chemistry at the University of North Carolina at Chapel Hill. Today, I'm going to be briefly telling you about a project that was funded by the North Carolina Space Grants, where we are trying to elucidate the redox mechanisms of extremotolerance in tardigrades. Now, tardigrades, also known as water bears, are microscopic invertebrates. They're only 100 to 200 micrometers long, but they have a full nervous and digestive system. Although they don't have a respiratory or circulatory system, instead they have what are known as storage cells. These globule cells you see pictured here that are able to float throughout the organism and provide needed nutrients to the internal organs. They also have a brain and what's known as eye spots, which are eye-like structures that enable the tardigrades to find their food in their environments. Now, what's interesting about tardigrades is their placement on the geological timescale. So here we have time on the x-axis and we have the number of genera on the y-axis. So over here in the Cambrian explosion, you see the sudden increase in overall genera on Earth. But eventually you have 1, 2, 3, 4, 5 mass extinctions between then and the present. Tardigrades, however, evolved close to 600 million years ago, which means that they have predated all of these known extinctions events. Not only have they survived, but they have also thrived. There's over 1,300 known species of tardigrades, and they have been found on every continent on Earth. So the secret of their survival lies in their ability to undergo something called cryptobiosis, which is an ametabolic state that can last from several hours to several years. And what's different about tardigrades as compared to other species capable of cryptobiosis is the ability to do this from a variety of different stressors. So tardigrades are able to undergo anhydrobiosis through desiccation, cryobiosis through freezing, anoxybiosis through oxygen depletion, chemobiosis through chemical exposure, osmobiosis through high osmolarity, and UV-induced diapause through irradiation. When they go into their tun state, you can see it pictured here, they bring their limbs into their body, expel their water stores, and kind of scrunch down, becoming a fraction of the size that they started. When they're in this particular state, they're even stronger and able to survive even more extreme stressors than when they are in their active state. There are several proposed mechanisms of tardigrade survival, including water replacement, vitrification, molecular shielding, and antioxidants. However, nobody knows really what signals them to go into that tun state that we saw before. And that's what my lab is trying to figure out. So to do so, we're using a species called Hypsibius exemplaris, imaged here, and they are relatively easy to culture in all female asexual species, and they've been fully sequenced multiple times, which is particularly good for proteomics. Their transparent body aids investigation, especially microscopic studies, and there's already an established molecular tool chest, which we can leverage for biological investigations. Our first step was to optimize a global protein analysis. So we used a recently published speed extraction, shown here on the left, which avoids detergent to use an acid-based extraction. And we have found that this is rapid, clean, and reproducible, and able to allow us deep coverage of the tardigrade proteome. After that, we decided to make sure that we could use our known Redox platform in tardigrades. So we did our extraction, and as you see our Redox platform here, we applied this to the tardigrade proteome, and we were able to identify over 1,400 redox sites on almost 500 proteins, which shows that we're going to get the deep coverage we need to understand how redox chemistry plays a role in initiating cryptobiosis. So our next steps are going to be to take these platforms that we've been able to optimize, combine it with physiological characterization that is being conducted through our collaborators at Marshall University. And together, we'll believe that we will have a comprehensive framework for cryptobiosis initiation within tardigrades. Before I finish, I'd like to acknowledge my lab, particularly my advisor, Dr. Leslie Hicks, the North Carolina Space Grant and the National Science Foundation, as well as the Coling Lab at Marshall University in West Virginia, who has been collaborating on this project.